All right, so the article is erythritol and blood clotting, and it looks at the new study in Nature Medicine that was brought to us by the Hazen group at the Cleveland Clinic. These are the same folks bringing us the TMAO research. And what they found, I'm actually going to skip down to my more detailed bullet point of what they found. So what they found was in two separate cohorts with a high prevalence of diabetes and adiposity, plasma erythritol was higher among those with cardiovascular disease and major adverse cardiovascular events. Of course, erythritol is a sweetener that a lot of low-carb foods are using. And so the thrust of this paper is maybe the erythritol that you're using as a sweetener is going to kill you of cardiovascular disease. All right, so they find the correlation, and they also incubate platelet-rich plasma or isolated platelets with erythritol, and that made the platelets more likely to aggregate in response to other stimuli that are routinely used for platelet activation assays in vitro. But glucose and 1,5-anhydroglucatol did not do this. 1,5-anhydroglucatol is a polyol that is used as an inverse marker of glycemic control. We'll get to what a polyol is in a moment. They also showed that human blood was more likely to show platelets adhering together when subject to shear stress if erythritol was added to it. And so note that these are a collection of in vitro studies using human cells and platelets. But then they also do a mouse model of carotid injury. The carotid artery is the artery that feeds the brain and can get blocked to cause a stroke. And this study took place over the course of 8 minutes after they injected the mice with erythritol or saline or 1,5-anhydroglucatol. And the blood appeared to clot faster when the mice were injected with erythritol just prior to the injury over the course of 8 minutes, but not when they were injected with saline or 1,5-anhydroglucatol. And then they gave people a drink, humans, this is in vivo, they gave people a drink with 30 grams of erythritol and showed that the erythritol remained elevated in the blood for 2 days, and it remained above the clotting thresholds from these other experiments in the other bullet points for more than a full day. So roughly a day and a half, the plasma erythritol after 30 grams of an erythritol drink is above the threshold required in these in vitro and mouse experiments to cause an increased propensity for clotting. All right. In order to make any sense of this, we need to go back and look at two pathways. One is the polyol pathway, and one is the pentose phosphate pathway. So polyols are, very broadly speaking, in organic chemistry, the suffix refers to an alcohol, which is a hydroxyl group or an OH group, oxygen and hydrogen atoms bound together, that is bound to an organic molecule. And a polyol is a compound with multiple OH groups. Now, by that definition, all the sugars are polyols. But in biochemistry and food science, we use the term a little bit differently, where a polyol tends to refer to a more specific subset of polyols known as sugar alcohols. And we distinguish sugar alcohols from sugars in that even though sugars are alcohols, right, because they all have OH groups, when we talk specifically about a sugar alcohol and distinguish it from a sugar, the sugar alcohol has all of the carbons, all of its oxygens are participating in OH groups. Whereas in a sugar, we have at least one carbon. So for example, this is glucose right here. We have at least one carbon, and here it's carbon 1 at the top, that is participating in a carbonyl group, which is carbon double bonded to oxygen. And the carbonyl group relative to the hydroxyl group or OH group or alcohol group, all of which refers to one of these guys, the difference is that the OH group is more reduced and the carbonyl group is oxidized. Things that are oxidized have fewer electrons. You see that by them having fewer hydrogens and or more double bonds. And so C double bond O, carbonyl group, is oxidized, it's lacking hydrogens, and it has a double bond. OH group is reduced, it does not have a double bond, and it has more H's. Right. So what you see here is glucose, and when the carbonyl group is at the end of the molecule, it's an aldehyde. And when the carbonyl is in the middle, meaning not on the end, it's a ketone. Glucose has an aldehyde group, and so ose means it's a sugar. So glucose is a hexose because it has 6 carbons. So 6 carbon sugar is a hexose. It's an aldose because it has an aldehyde group, and it is an aldohexose. This is fructose, and fructose, or excuse me, sorry, this is galactose. So galactose is an isomer of glucose. The difference is that this carbon 4 has, in this depiction, glucose has it on the right, galactose has it on the left. It also is an aldohexose. This is fructose. It's got a carbonyl not on the end. In between some of the carbons, that's a ketone carbonyl. Therefore, fructose is a ketone. It's a ketose because it's a ketone sugar, and it's a ketohexose because it's a ketone 6 carbon sugar. Um, I think I put the wrong. I think I put the wrong structure here. So we'll skip over the structure. In any case, one of the sugars that we'll talk about is erythrose, and erythrose is an aldehyde. It's an aldose, but it's a 4 carbon sugar for it. So it's a 4 carbon sugar. So therefore, it is a tetrose and a aldotetrose. All right. So the polyol pathway is a pathway where we can reduce sugars to their corresponding alcohols. And as 3 examples, that pathway would reduce glucose to sorbitol, galactose to galactitol, and erythrose to erythritol. Generally, the polyol pathway is a way of disposing of excess sugars that go beyond a certain threshold. It is a backup mechanism. It is not something that you are trying to engage in, but my suspicion is it is solving 2 problems. One would be a big one would be the fact that carbonyl groups are very reactive, and especially aldehyde groups. And so you do not, if you have an excess of sugars that you cannot metabolize through the energy metabolism pathway and do something useful with, you want to protect them from engaging in glycation reactions. So I think that's one reason why you would want to reduce them to polyols. Another reason that you would want to reduce them to polyols, I think, is because polyols tend to accumulate water and they tend to push themselves out into the urine. So if you take 75 grams of erythritol, over the course of 24 hours, you're going to lose 78% of it in your urine. And so I think from the perspective of why would a human cell want to convert glucose to sorbitol under conditions of hyperglycemia, I think part one of it is get rid of the reactive carbonyl group. And then second of it is, to the extent these things get pushed outside cells, they're going to create an osmotic effect that will drive water pressure to push them out into the urine. And the reason that they hold on to water so well, and one of the negative properties of them, is they can contribute to osmotic stress because, and we'll see the importance of this momentarily, sugar alcohols do not readily cross membranes through sugar transporters, and therefore they tend to get stuck on one side of the membrane and draw water unto themselves without the cell being able to respond by equilibrating their concentration across the membrane and spreading out the forces attracting the water. And osmotic stress can, if it's outside the cell, cause, meaning if this impermeable solute is outside the cell, in this case the sugar alcohol, cause cell dehydration and shrinkage. And if it accumulates inside the cell, can cause cell swelling, either of which can damage the cells. Now, the other problem with the polyol pathway is that generally this is mediated by either the enzyme aldose reductase or some other enzyme within the family of aldoketoreductases, all of which use NADPH to reduce the sugar to the sugar alcohol. The reason that's a problem is that NADPH is also used for the recycling of vitamin K and folate, the recycling of glutathione, the detoxification reactions of cytochrome P450 enzymes, and the anabolic synthesis of many things. But precisely in this recycling area with glutathione, NADPH is supporting the entire antioxidant system. You know, taking us back to the previous discussion about hair graying, sapping NADPH is going to be terrible for your antioxidant system, and therefore the premature graying of hair. Now, as a result of these potential solutions to problems, but also negative implications of the polyol pathway being activated, that is especially so osmotic stress and sapping NADPH, you use this as a backup mechanism only when you have spillover effects of the sugars. And so I'll give you some data supporting that. So what you see here is plasma glucose on the horizontal axis plotted against cerebrospinal fluid sorbitol on the vertical axis in three groups. One is healthy individuals, one is individuals in green with elevated CSF glucose, and then in purple, the healthy individuals are the blue circles. And the purple diamonds are individuals with type 2 diabetes. And you can see there's this cluster of sort of normal sorbitol levels, and then there's this kind of gap up that starts occurring around 8 millimolar glucose, which is 144 milligrams per deciliter. So the sorbitol rising above normal is a spillover effect of glucose elevation over 140, basically. And in support of that, there is a study showing that in diabetics, if you use insulin to decrease their glucose level to under 140, you get their otherwise elevated sorbitol levels to drop to normal, to the level of normal healthy controls. And it's also the case that sorbitol levels in red blood cells in healthy people and diabetics together inversely correlate with glutathione, which is supporting the framework that for activation of the polyol pathway and the conversion of glucose to sorbitol as an escape valve of excess glucose under conditions of hyperglycemia taxes the supply of NADPH needed for glutathione recycling and thereby causes oxidative stress. Now, here's another study where they took two types of people. They looked at what they did was they gave them a galactose tolerance test. So galactose is the sugar that you get when you digest milk sugar, which is lactose. You get glucose and galactose. And tolerating milk isn't all about lactose. It's also about galactose. And what you're seeing here is the people on the left, group A, had very high galactose peaks after a galactose tolerance test. And the values are milligrams per liter. So if you convert that to milligrams per deciliter, you divide it in 10. So this is peaking 40 to 50 milligrams per deciliter. Whereas on the right, you have the galactose tolerant subjects who were peaking under 30 milligrams per deciliter. And then they looked at the galactitol, which is the sugar alcohol made from galactose during the activation of the polyol pathway, analogous to the conversion of glucose to sorbitol. And you can see that the blood galactitol on day one hit 7.4. Then it was 5 times lower after a week of consuming galactose every day. It only went to 1.5, and then on day 15, 0, just like the galactose tolerant group. So what that's showing you is that in certain subjects who did not have high enough expression of galactose metabolizing enzymes, they had greater galactose peak in plasma because it was not going all the way down, being burned for energy. And because it wasn't going all the way down burned for energy, what galactose did make it into the cell was largely spilling over into the polyol pathway to reduce the galactose, or not largely, but some of it was spilling over to the polyol pathway to reduce the galactose to galactitol. So presumably we have something analogous where erythros is converted to erythritol endogenously. But to understand that and why it would happen, we need to look at the pentose phosphate pathway. So the pentose phosphate pathway, and this slide is from my this slide is from my energy metabolism course, lesson 27. All right, so the pentose phosphate pathway operates as a shunt from glycolysis. A shunt is taken from electricity, electronics, where a shunt is something that comes out from a circuit, goes back in. So it's also called the hexose monophosphate shunt, because these hexose phosphates are shunted into the shunt. It's called the pentose phosphate pathway because the 6-carbon hexose phosphates are converted into 5-carbon pentose phosphates. Now, there's two purposes of this. One is to produce NADPH. And when I say produce NADPH, I mean take NADP plus and load hydrogen ions, electrons, and their energy onto it to make NADPH. So you're loading up energy, electrons, and hydrogens as passengers on the NADP plus bus, so they can ride the bus as NADPH into other pathways. And then you also get 5-carbon sugars, and 5-carbon sugars can then be used for structural purposes. Now, quite often, the cell doesn't need as many 5-carbon sugars as it needs NADPH, and so the pentose phosphates, rather than being used for structural purposes, can be rearranged into 3 pentoses can be rearranged into 2 hexoses and 1 triose. The hexoses go back to glycolysis at the level of hexose phosphates. The trioses go back to glycolysis at the level of triose phosphates, where what's happening in the middle is you're splitting apart 1 hexose phosphate into 2 triose phosphates, right? So if what you need is NADPH, you just run this in a loop, an endless loop. And of course, you're losing 1 carbon every time you do this, but you can run this 6 times, use all 6 carbons from the hexose, and get 8 NADPH out of it, rather than taking 1 whole hexose, get 2 NADPH, and have a 5-carbon sugar leave for structural purposes, right? This is based entirely on a need-based pathway. If you need 5-carbon sugars, the concentration outside the pathway declines because you're running out of them, and so therefore the spillover leaks out of this pathway into that pathway, and the enzymes that utilize them are missing substrates, and they'll help drive that the loss of 5-carbon sugars from this pathway into the other pathways forward. Similarly, the need for NADPH is what drives this part forward in the form of NADP+. When you have more NADP+, that means you need more NADPH. That's why you have more NADP+. If you have more NADP+, you have this reaction going forward at a greater rate because there's more substrate available for that reaction. This right here is called the oxidative phase. The non-oxidative phase is the rearrangement of sugars whose sole purpose is to take the pentose phosphates and recycle them through glycolysis to run this as a loop, to use all of the carbons within any given hexose entirely for NADPH production rather than 5-carbon sugar production. Now, and these 5-carbon sugars, by the way, some of the structural purposes they're listed right here. NADPH itself, it has 5-carbon sugar structural component. So does NADH, FADH2, CoA, and ATP, DNA, RNA. Those are some of the uses why you would need those. Okay. Now, in the process of rearranging the sugars to go back to glycolysis, you produce, through the action of transketolase and then transaldolase, erythros 4-phosphate, which is a 4-carbon, it's a tetrose phosphate, a 4-carbon sugar bound to phosphorus, or to phosphate rather. And to help clear this, to finish going through glycolysis, you need to use transketolase again. there's two enzymes here, transketolase, transaldolase. Transaldolase has no nutritional cofactors. Transketolase has one nutritional cofactor, thiamin, vitamin B1. So why would you get an accumulation of erythrose 4-phosphate under any conditions? Probably because you're thiamin deficient. You're not so deficient that you have no transketolase activity, but you're deficient enough that you engage transketolase once and transaldolase once to get erythrose 4-phosphate, and you don't have enough transketolase activity to efficiently and rapidly convert all the erythrose 4-phosphate into the 6 and 3 carbon sugars of the next step to go back to glycolysis. So the fact that your transketolase activity is mediocre as a result of suboptimal thiamin status is probably the one and only one. Well, I shouldn't say the one and only reason, because you can have damage to the enzyme, you can have a genetic polymorphism in the enzyme, you can have, chronic alcoholism creates epigenetic suppression of the enzyme, right? But from a vitamin nutritional standpoint, the one thing relevant is thiamin, and then the transketolase is the one thing that's enzymatically relevant. You could break that into several possible things that could go into this, but mediocre transketolase activity, possibly as a result of thiamin deficiency, is really the one thing that could make you accumulate erythrose 4-phosphate, because your purpose in making it is not to produce erythrose 4-phosphate. It's an intermediate in the conversion of 5 carbon sugars when you don't need them to 6 and 3 carbon sugars, so that you can run this in a loop and make more NADPH, right? So your activation of this pathway is robust, but your ability to complete the pathway sucks. So you are sloppily activating the pentose phosphate pathway if you have an accumulation of erythrose 4-phosphate. Now, you will not find in a biochemistry book, so I have the biochemistry book by Berg and Stryer, the latest edition of it. Search erythrose, you will find a bunch of hits. Search erythritol, 0. Search erythrinate, 0. But what happened was, in a series of studies over the last decade, or somewhat less than that, plasma erythritol has been shown to be elevated in diabetics who develop retinopathy versus diabetics who do not. Those who have recently developed central adiposity, meaning visceral belly fatness, versus those who have had a stable level of body fat, and people who have new onset type 2 diabetes. And these are huge differences in erythritol, like 15-fold differences in erythritol or 21-fold differences in erythritol. So 15-fold differences in plasma erythritol between people with incident central adiposity versus stable adiposity. 21-fold differences in plasma erythritol between people who had HbA1c over 5.05% and under 5.05%. So what is this signifying? Well, if we go back to the graph, why would you have think about metabolic dysfunction? What's going on that's going to lead to erythrose 4-phosphate accumulating? You have robust activation of the pentose phosphate pathway. Why would you do that? It is not from a push forward effect of glucose. That is not how this pathway is regulated. This pathway is regulated by two things: the need for 5 carbon sugars and the need for NADPH. Well, what ties metabolic dysfunction with the need for NADPH? Hyperglycemia, conversion to sorbitol, activation of the polyol pathway, saps NADPH. Boom. You have lots of NADP+, you need to make NADPH. De novo lipogenesis from insulin resistance. Boom. NADPH is used up in the synthesis of fatty acids. Now you need more. Activate the polyol pathway, you get oxidative stress from NADPH depletion. What do you need to do with NADPH? Recycle glutathione. What does that do to your NADPH? It uses it. What does that do to the pentose phosphate pathway? It activates it. Activating the pentose phosphate pathway should be a solution, not a problem. But if you're thiamin deficient, or you're an alcoholic, or you have a genetic polymorphism in transketolase, you are going to get this sloppy activation of the pentose phosphate pathway that is not going to meet your needs for NADPH because you're not fully looping it back the way you should, and instead you have a spillover of erythrose 4-phosphate as one of numerous intermediates that's being reduced to erythritol. Now, when those findings were made, it was a bit of a puzzle because there's no textbook that says that you're supposed to make erythritol from erythrose 4-phosphate. But follow-up studies were done using isotopic labeling in humans, showing that erythritol is a metabolite of the pentose phosphate pathway. What does that mean? That means that when you do not complete the pathway you should because you've sloppily activated it and you're thiamin deficient or otherwise transketolase deficient, you have erythrose 4-phosphate being built up faster than can be metabolized back to hexose phosphates and triose phosphates to loop through glycolysis, and therefore you dephosphorylate it to erythrose and you reduce it to erythritol. A 2019 paper, that was done in 2017, a 2019 paper came out and showed that erythrose can be reduced to erythritol using the enzymes alcohol dehydrogenase 1 and sorbitol dehydrogenase, again, using NADPH as we would expect. But again, what does that mean? That means that not only are you not completing the loop to effectively generate NADPH, it means now you're using it. Now your sloppy activation of the pentose phosphate pathway has created an NADPH sucking liability in the form of spillover of intermediates, which saps your NADPH further and further activates the pathway in a vicious cycle that loops out of control the production of intermediates that activate the polyol pathway in spillover. No wonder you can have something like HbA1c 5.05% above and below 20-fold difference in erythritol, because you're not talking about a linear, like, you're not talking about a case where there's always some erythrose 4-phosphate and there's always some spillover into erythritol. You're talking about a threshold effect that activates a vicious cycle and loops out of control in the onset of metabolic dysfunction. Now, interestingly, the question arises, can erythritol sweeteners be utilized in the pentose phosphate pathway? Well, so you're telling me erythrose 4-phosphate can go out to erythritol, but erythritol can't go back into in the opposite way? Doubtful. Very doubtful. So that 2017 study that showed erythritol is produced by the pentose phosphate pathway showed that 5 to 10% of supplemental erythritol is metabolized to erythrinate. That means that it is oxidized to a carboxylic acid in an analogous way to how glucose 6-phosphate with glucose 6-phosphate dehydrogenase and lactinase in the first oxidative step of the pentose phosphate pathway is converted to 6-phosphogluconate. In other words, 6-phosphogluconic acid. Now, that would seem to suggest that erythritol can be oxidized to erythrose and phosphorylated to erythrose 4-phosphate, which maybe is the substrate for the production of 4-phosphoerythrinate, which is then hydrolyzed to erythrinate in an analogous way. But they didn't identify the enzymes involved, so we don't know that it happens in exactly that way. That's just my guess. But I think that no one was looking for erythritol production in humans until they looked for it. And I think that, had they looked for utilization of erythritol in humans in the pentose phosphate pathway, they might have found it. Instead, they looked for erythrinate production, which there was no reason to believe existed in 2016, and they found it in 2017 because they looked for it. And they said 5 to 10% of erythritol is converted to erythrinate, and they assumed that most of the rest went into the urine. Go back to the previous papers from the 90s that show that, they do these you can't measure the absorption of anything if you don't measure the fecal contents left over. This seems obvious to me, but indicts almost all absorption studies as fallacious. But anyway, they showed that 78% of the erythritol turned up in the urine within 24 hours unmetabolized, and they said, well, that means 20% of it was available to the microbiome for short-chain fatty acid production. Are you kidding me? You didn't measure fecal erythritol content. You don't know that. So now, more than a decade later, someone comes out and says, well, actually 10% of it was metabolized to erythrinate. Why wasn't 10 or 15% of it metabolized in the pentose phosphate pathway? My suspicion is it was. Now, anyway, so this new erythritol study, what do they show? Well, the first thing they show is that in 2 cohorts with a high prevalence of diabetes and adiposity, plasma erythritol was higher among those with cardiovascular disease and major cardiovascular events. I mean, what does that mean? After everything that we just looked at, it means these are the people with a sloppily activated pentose phosphate pathway due to hyperglycemia activation of the polyol pathway sapping NADPH, due to insulin resistance causing de novo lipogenesis sapping NADPH, due to oxidative stress from other problems, or from sapping NADPH in these first 2 areas, that's sapping NADPH, all of which is activating the pentose phosphate pathway even more, but it's sloppily activated because their transketolase is deficient, probably largely due to thiamin deficiency, perhaps due to genetic polymorphisms in transketolase or alcohol, or epigenetic effects of alcoholism or whatever, but quite often from suboptimal thiamine status. Their activation is suboptimal. They have spillover effects from erythrose 4-phosphate and other intermediates that lead to the further activation of the polyol pathway because of the activation of the pentose phosphate pathway, which leads to more NADPH depletion, which leads to more activation of the pentose phosphate pathway, which can never overcome the limiting factor being the transketolase, which then it leads to a metabolic deterioration in a vicious cycle that just leads to ever more NADPH depletion. Is the first thing I would go to, well, these people must have been eating keto snacks that were sweetened with erythritol? No. Did they measure erythritol intake? No. Granted, they did say that it's difficult to measure because FDA hasn't mandated that keto snacks disclose the grams of erythritol in their snacks, but they didn't ask people if they were eating keto snacks either. They made no effort to measure in any capacity the intake of erythritol and have no basis whatsoever for indicting dietary erythritol at all. Okay, then they incubate platelet-rich plasma or isolated platelets with erythritol, and this made the platelets more likely to aggregate in response to other stimuli, but glucose and 1,5-anhydroglucotol did not do this. Now, 1,5-anhydroglucotol is a polyol, but it very closely resembles glucose. It occurs in food. That's where you get it. And it is thought to be an inverse marker of glycemic status because when glucose goes above 180, the kidneys have trouble reabsorbing it. And so the level of 1,5-anhydroglucotol in the blood will decline when you are exposed chronically to glucose above 180 milligrams per deciliter. Then they took human whole blood and found it was more likely to show platelets adhering in response to shear stress if erythritol was added. And then they took a mouse model of a carotid injury over the course of 8 minutes, and the carotid artery occluded faster when they were injected with erythritol, suggesting faster clotting. And then finally, they showed consumption of a drink with 30 grams of erythritol by human volunteers elevated erythritol beyond the levels needed to cause clotting in vitro and to cause clotting in mice, stayed elevated for 2 days. For about a day and a half, it stayed elevated above those thresholds. So they are saying they have this clean story where you eat erythritol, your erythritol goes up that much, it does all these clotting things. But I think this is very misleading. And they made no effort to lay out the mechanism by which erythritol causes clotting, so I'm going to do it for them. And I'm not saying that I know this is what happened, but I am saying that they didn't offer a mechanism, so it behooves us to come up with a mechanism, and it makes sense to fit erythritol into the most generic known thing that we know it would do, that we know causes platelet activation. So in 1996, researchers were trying to determine why some types of contrast media were causing platelet aggregation. So they mixed together various ingredients to make solutions that could control for osmolality and ionic strength. Osmolality, which is very similar to osmolarity, they're just defined a little differently, is a measure of the number of dissolved solutes per unit of mass of solvent. Osmolarity is per unit of volume of solvent. It's if the solvent is water, it's almost the same thing. But this is referring to the number rather than the mass of the solute, right? So if you have a big molecule, small molecule, it doesn't matter. It just matters how many of the solutes are there per unit of mass of the solvent. And normal osmolality is around here. It's 260 to 330. 260 to 320 milliosmoles per kilogram solvent. That's normal plasma variation, and that's normal cell culture medium. And that's right around their starting point. And you see that as you go a little bit above that, you have this dramatic escalation in platelet activation from increasing osmolality. Huge effect, very tight in the range near normal. And then there's this very small effect of ionic strength, whereas the ionic strength decreases, you have increasing platelet activation. Sorry, this on the horizontal axis, ionic strength increases, showing that on the vertical axis, platelet activation decreases as you increase in ionic strength. And so how do we relate that to erythritol? Well, erythritol is a sugar alcohol. All sugar alcohols are known to cause osmotic stress. This is well known, and that's because they don't cross membranes very easily. So if you compare the transport of glucose versus erythritol, erythritol has 1,000 times slower transport of itself across a cell membrane in red blood cells compared to glucose. If you look at 1,5-anhydroglucatol, it is half of glucose. So glucose and 1,5-anhydroglucatol are not going to cause osmotic stress at the same concentrations as erythritol, because the cell can just move the glucose or the 1,5-anhydroglucatol into itself. So if the glucose or 1,5-anhydroglucatol is dry in water, the cell does not need to worry about the water differential across its membrane causing osmotic stress, because it can just move the glucose or 1,5-anhydroglucatol into itself. But it cannot do this with erythritol fast enough, because the transport of erythritol is 1,000 times slower than glucose. Now, if you look at the concentrations of erythritol used in the study, it's in the area of 45 to 270 micromolar. Now, that micromolar is equivalent to the milliosmoles, right? 1 micromolar something would, assuming your water is your solvent, so 1 liter equals 1 kilogram, you can essentially say 1 micromolar added would increase osmolality 1 microosmol, which is 1,000th of 1 milliosmol. And so, you look at this graph, and you can say, well, yes, I understand that it's precipitously increasing platelet activation, very close to the normal, to deviations above normal, but this deviation from here to here is at the level of more than 100 milliosmolar. And so, how is 45 to 200, 247 or whatever it was, how 45 to 270 micromolar, how is that going to have enough impact on that? And the answer is that in plasma, most of the osmolality is glucose and sodium. In mouse plasma, most of the osmolality is in glucose and sodium. All of their experiments were done either with human blood or with live mice, where the primary osmolytes are glucose and sodium, or they were done with isolated platelets or cells that were incubated in Hank's salt buffer solution, which has glucose and various mineral salts, including sodium, as the primary osmolytes. And these all easily go in or out of the cell when the cell wants them to. And one of the things that the cell does to regulate them is defend itself against osmotic stress. So you can't look at that graph and translate those number of millimoles osmolality changes to what you need to get platelet activation when erythritol is unique in its inability to rapidly equilibrate across the membrane. Not unique. This is a unique property of sugar alcohols. So what you would expect is that you have this, 1,000-fold greater potency of erythritol in causing osmotic stress, and therefore you don't need very much you don't need even 1 millimolar osmolality change. You just need this intransigently non-permeable solute in small amounts that will cause the osmotic stress that will activate the platelets. Now I can't prove that with a study because the Hazen group at the Cleveland Clinic, who brought us this paper, are the first to look at the effect of erythritol on platelet activation. But they didn't offer a mechanism. So therefore, if we know that erythritol causes osmotic stress because all sugar alcohols do, and that osmotic stress activates platelets, why do we not just fall back on that as a working model? And if we fall back on that as a working model, then this totally invalidates everything that they did, because when you consume erythritol, what is very likely to happen is that your physiology will regulate osmolality for its own sake in your blood. So if erythritol elevates the osmolality because it doesn't efficiently equilibrate across the membrane, your kidneys and other organs are going to change the concentrations of glucose and sodium in your blood to match it. And you don't have to make a big change in them. Even if you dealt with 270 micromolar erythritol exclusively by decreasing plasma glucose, you would only have to decrease it by 5 milligrams per deciliter to completely account for the change in osmolality induced by the erythritol. And a change of 5 milligrams per deciliter of glucose is trivial in terms of the glucose result. This is very well-established textbook physiology that's known, for example, that if you have hyperglycemia, that can cause hyponatremia, meaning low blood sodium, because if you have too much glucose in the blood and the body can't deal with the glucose effectively due to an insulin problem, your kidneys will pee out the sodium to regulate the osmolality of the blood. And, if you feed rats erythritol, they do lose more solutes in their urine. Granted, they also drink more water and pee out more water, and no study that I've seen has looked at how plasma osmolality has changed. But I bet you anything, plasma osmolality remains relatively stable as a result of the physiological adjustment to specifically regulate osmolality of the plasma for its own sake. And so if elevated plasma osmolality is going to activate platelets, your body is probably going to try to not have plasma osmolality elevate. And so therefore, what would you want to see? What you would want to see is that they fed people 30 grams of erythritol, allowed the physiological adjustments to take place, then took their blood and showed that the platelets could be more easily activated. They didn't report doing that. I suspect that they did do it, and they didn't like the results they got, but they did not report doing that. They reported doing all the experiments by adding erythritol to healthy non-erythritol supplemented blood, showing that causes the clotting, and then feeding people erythritol, then drawing their blood and showing the concentration of erythritol in that blood is more than what was needed in the experiments using the blood of the people who was not erythritol supplemented. So you're telling me that they had to draw blood twice, once to supplement people with erythritol and once to supplement the blood with erythritol to mimic feeding people erythritol. You're telling me they couldn't just test the erythritol supplemented human blood for clotting? You're telling me they drew the blood anyway. So I find it borderline obvious that they did do that test. They didn't like the results, and so they only reported the results of adding erythritol to the blood, because it just makes no sense that they wouldn't just clinch this by showing that the blood of the people that they drew anyway, who ate the erythritol had increased clotting. And this, by the way, is what they did for TMAO in 2017. They showed that if you supplement people with choline, they have more TMAO in the blood, which, by the way, the major physiological function of TMAO is as an osmolyte. They had more TMAO in the blood, and they showed that ex vivo, their blood did clot, right? So my concern about TMAO is a 3, like I've said before. My concern about erythritol causing blood clotting and cardiovascular disease from this study is a 0.1, because they didn't do even that. But again, it looks like they must have done that, not like the results, which makes me deeply suspicious about their selectivity in reporting, which makes me pay even less attention to this study. So, bottom lining this, I think it's misleading the way they single out keto food sweeteners. Plasma erythritol likely serves as a marker of sloppily activated pentose phosphate pathway, indicating NADPH depletion and suboptimal thiamin status. The NADPH depletion is caused by oxidative stress, hyperglycemia, activating the polyol pathway, and inflammation and insulin resistance, increasing de novo lipogenesis, as well as the vicious cycle that you get from the oxidative stress involved in all of that, and then also the NADPH depletion that is caused by further activation of the polyol pathway by the activation of the pentose phosphate pathway exceeding the thiamine-dependent transketolase enzyme, thus creating a vicious cycle of progressive greater activation of pentose phosphate pathway, inability of thiamine transketolase to keep up, and therefore spillover effects more and more into the polyol pathway in a vicious cycle that will cause metabolic deterioration. I think that's what's happening when plasma erythritol being 15-fold, 21-fold elevated is showing the transition states. And notice, by the way, that those studies showing erythritol being that elevated are at the transition points. It's at new onset central adiposity versus stable adiposity, new onset diabetes, diabetic retinopathy versus diabetes, but stable controlled diabetes versus healthy control doesn't have the elevation of erythritol. So I think this is a precipitous metabolic deterioration at the transition point into disease is what this erythritol is acting as a marker of. In a lot of these studies, maybe it's also a linear indication of cardiovascular risk as well. But I think these previous studies of the massive elevation of 15-20-fold, I think that's acute metabolic deterioration. In this Hazen study, I think they're showing a more moderate linear effect, but it's still the same thing. It's oxidative stress, polyol pathway from hyperglycemia, inflammation, insulin resistant, activating de novo lipogenesis, all of those depleting NADPH, activating the pathway further, not having the thiamine-dependent transketolase step keep up. The pro-clotting effects shown in vitro in mice were probably reflecting osmotic effects of erythritol that would play out in a healthy human eating erythritol over the course of a day, but in the case of the mice, their physiology couldn't adapt because this study was 8 minutes long. And so, bottom line is I don't use erythritol, but if I were to consider using erythritol, this study would play no role in my decision.